Methods of molecular biology. We want introduce today the genetic diversity of viruses. This is a lecture note. In 1898, Martinus Beijerinck, a professor of microbiology at the Technical University at Delft, the Netherlands, put forth his concepts that viruses were small and infectious. He defined the infectious agents excuse me, as a contagium vivum fluidum. It was a small, and after he discovered that the virus readily passes through a porcelain filter, suggesting that it was smaller than bacteria. Resembles 1892 experiments of Dmitry Ivans Ivanovsky. He also observed that the agent could diffuse through agar that retained bacteria, and furthermore that the virus couldn't be cultured except in living growing plants. The virus was tobacco mosaic virus (TMV). This report suggesting that microbes need not be cellular was to forever change the definition of pathogens. Bacteriophage, Bacteriophages played an essential role in the development of molecular biology early at the discovery of the genetic role of nucleic acids, and more recently with the discovery of CRISPR-Cas system that became the basis of the genome editing revolution. The amount and the arrangement of proteins and nucleic acid of viruses determine their size and shapes. The nucleic acid and proteins of each class of viruses assemble themselves into a structure called nucleoprotein or nucleocapsid. Some viruses have more than one layer of protein surrounding the nucleic acid, and still others have a lipoprotein membrane or envelope derived from a membrane of the host cell that surrounds the nucleocapsid core. Capsids protein interact by highly specific, flexible, non-covalent contacts. Penetrating the membrane are additional proteins that determine the specificity of the viruses to host cells. To a host cell. Basic morphology of the viruses is presented on figure 2. The figure 2 representative virus showing a range of morphologies. Tobacco mosaic virus or Thomas Moravec and G1 cluster mycobacteriophage or Brainford megavirus Chantral abevage and maize. D is a maize. Okay, number A is a tobacco mosaic virus. Number B is a cluster mycobacteriophage. C is a megavirus. D is a maize streak virus. D. E is a holovirus. His number 1. F is a comation latent virus. F is a comation. G is a Ebola virus. G. H is a poliovirus. is a poliovirus, and I is a avian influenza H7N9. Long terminal extension and loops make each viral capsid unique. Virus capsids are essentially molecular containers. What do they contain? What is the charge of the contents? Viral genomes may consist of either double or single strand DNA or double or single stranded RNA. Depending on its type of nucleic acid, a virus is called a DNA virus or an RNA virus. Double or single stranded DNA, double or single stranded RNA. Okay, figure 3 is a taxonomy and replication strategy of RNA virus. Note, A is a simplified taxonomy of the genome architecture of the RNA virus, and the B is an infection with the plus or positive RNA virus exemplified with the co-virus or COVID-like virion, and it's available also in the Wikipedia. Okay, now we want to go through this. You see, we have an RNA. The RNA is a desoxyRNA or ssRNA. If you have a desoxyRNA, well, it's maybe a BMAL, IBDV, cysto, sigma 6, reo, the rotavirus or reovirus, or it will be a ssRNA. ssRNA, it could be a positive RNA from the family of Boromovirus. For example, virus is a BMV. Nido family, for example, SARS, CoV, PRRSV, EAV virus, MERS virus or CoV, Picoma virus family, PV virus, for example, FMDV virus or EV71 virus from the family of Calici, for example, MNV virus or MV virus, Tombus virus family, for example, virus TCV, Alpha virus family, for example, virus CHIKV or SINV, for example, Flavivirus family, YFV, WNF, DV, GEV, BVDV, and HCV. Retrovirus family, for example, HIV-1. In the negative RNA virus, we have a seg and non-seg. In the seg, we have a orthomixo, IVA. We have a bunia, for example, LCEV and SBV. We have RNA, for example, MACV and LCV. In the non-seg, we have a paramixo, for example, MV and SEV, and we have a rhabdo, for example, VSV. In the figure B, it's shown positive RNA virus, retrovirus with the capsid matrix, and negative RNA virus and desoxyRNA virus. It's very difficult for me now to understand the difference. I just noticed that the positive RNA virus can directly go to the transcription. I notice. As we know, DNA or deoxyribonucleic acid is the blueprint of life, coding gene. responsible for certain traits. Its double helix structure was established by James Watson and Francis Crick based on images produced by Rosalind Franklin in 1953. It is made of repeating subunits called nucleotides, which consist of phosphates, deoxyribose or sugar, adenine, thymine, guanine, and cytosine bases, connected by the hydrogen bonds. RNA instead is a single-stranded, contains ribose and uracil instead of thymine in the figure 4. In the figure 4, we have a difference between DNA and RNA. Okay, as we know, the main difference is a uracil thymine, and it's a double and single stranded. The other differences, I don't remember. I just remember it was a 5 differences. The protein and nucleic acid and constituents have properties unique for each class of virus. When assembled, they determine the size and shape of the virus for that specific class. On figure 5, we can see basis division of DNA and RNA containing viruses on enveloped and non-enveloped types. Non-enveloped viruses are composed only of capsid proteins, capsid protein and nucleic acid. Non-enveloped viruses such as rotavirus, adenovirus, and hepatitis A virus are more resistant than enveloped virus. Again, non-enveloped viruses are composed only of capsid and nucleic acid. Non-enveloped viruses such as rotavirus, adenovirus, and hepatitis A virus, rota, adeno, and hepatitis A virus are more resistant than envelope virus. More resistant than envelope virus like influenza virus, and not inactivated by agents affecting lipid, and not inactivated by agent affecting lipids such as alcohol, ether, cationic surfactants, and chlorhexidine. It's very difficult and important note. Non-enveloped virus such as rotavirus, adenovirus, and hepatitis A virus are more resistant than enveloped virus like influenza virus, and not inactivated by agents affecting lipids such as alcohol, ether, cationic surfactants, and chlorhexidine. For example, this is a HIV, HSV, HBV, and influenza virus. It's written here in the envelope virus, envelope nucleotide capsid and virion. And again, in the non-envelope, we have a norovirus, parvovirus, HAV, and HEV. It's written here envelope influenza, and again it's written here envelope influenza. It's again here written adeno, HAV is adeno, HEV is a Epstein-Barr, I don't know. HIV, HBV, HSV, and influenza is envelope. $15 million people die annually from infectious disease in the world. For instance, cervical carcinoma is caused by human papillomavirus type 16, 18, 31. Possible cause of diarrhea are Norwalk virus, rotavirus. Possible cause of diarrhea are Norwalk virus and rotaviruses. There are four types of influenza virus: A, B, C, and D. Human influenza A and B cause seasonal epidemic of disease almost every winter. A and B. The emergence of a new and a very different influenza A virus to infect people can cause an influenza pandemic. Influenza type C generally cause a mild respiratory illness and are not thought to cause epidemics. Influenza C generally cause a mild respiratory illness. Influenza D viruses primarily affect cattle and are not known to infect or cause illness in people. Okay, 1918 is a Spanish influenza, H1N1 influenza viruses. From the birth to human, transmission of H1N1 virus, hemagglutinin and neurotransmittase, all 8 genetic segments, although to have oriented from avian influenza viruses. 1957, Asian influenza, H2N2 influenza virus, H2N2 avian virus, and H1N1 human viruses. Reassortment, 3 new genetic segments from avian influenza virus introduced. HA, NA, and PB1 contained 5 RNA segments from 1918. Okay, in the 1961 Hong Kong influenza, H3N2, H3 avian virus, and H2N2 human virus. 2 new genetic segments from avian influenza virus introduced. HAPB1 contained 5 RNA segments from 1918. And next pandemic influenza is the avian virus or avian virus, H3N2 human virus. All 8 genes new or further derivatives of 1918. It's evolution of influenza A virus. Thus, viruses stimulate some great ideas in a structured dynamic function complex. In particular, understanding the complex function of a system by considering its components, thus possibility creating complex forms in biological system through self-assembly using a simplified model of the system to understand a wide range of more complex phenomena. Okay, there is also reference that's available here, and thanks very thanks from this Polytechnic University, and really very thanks from the Professor Aizan Zuzapova that's available, that's make this lecture note for us.